The science behind animal camouflage is fascinating and involves a variety of mechanisms, depending on the animal. While chameleons are iconic for their color-changing abilities, not all camouflaged animals share the exact same characteristics or mechanisms. Camouflage is a remarkable adaptation that allows animals to blend in with their surroundings, helping them avoid predators or ambush prey. This can involve matching colors, patterns, textures, or even imitating other objects. Camouflage generally works by manipulating how an animal's body reflects or absorbs light, thereby altering its perceived appearance to an observer, predator or prey. Here are the main scientific principles and mechanisms. Pigmentary coloration, biochromes. This is the most common form of animal coloration. Animals have specialized cells called chromatophores that contain different types of pigments, biochromes. That melanin produces black, brown, and some red/yellow colors. Carotenoids, often acquired through diet, producing yellows, oranges, and reds. Pterins produce yellows and reds. Flavins produce yellow colors. In these cases, color change, if any, often occurs more slowly via changes in pigment concentration or dispersion within cells like melanophores. Pigment granules can either concentrate in the center, making the animal appear lighter, or disperse throughout the cell, making it appear darker. This is influenced by hormones and the nervous system. Seasonal changes, driven by hormones in response to environmental cues like light and temperature, e.g., Arctic fox, snowshoe hare. Diet. Some animals derive their coloration from the pigments in their food, e.g., flamingos get their pink from carotenoids in their diet. Structural coloration. This type of coloration doesn't rely on pigments but on the physical structure of the animal's surface. Microscopic structures scatter or interfere with light waves, producing specific colors. Iridescence, caused by microscopic layers or prisms that reflect different wavelengths of light at different angles, e.g., peacock feathers, butterfly wings. Tyndall scattering/Rayleigh scattering, produces blue colors by scattering short wavelength light, e.g., the blue of a clear sky. Some blue frogs. Photonic crystals, highly organized, repeating nanostructures that selectively reflect specific wavelengths of light. This is a key mechanism for rapid, dynamic color change in some animals. Active versus passive camouflage. Passive camouflage, the animal's color and pattern are fixed and evolved to match a specific environment, e.g., leopard spots for dappled light, stick insects for twigs. This is background matching. Active camouflage, the animal can change its color, pattern, or texture in response to its environment or internal state. This is seen in chameleons, cuttlefish, octopuses, and some fish. Disruptive coloration, patterns on the body break up the animal's outline, making it harder for a predator to recognize its shape. Stripes, like zebras, or blotches often serve this purpose as mimicry. Masquerade, the animal mimics an uninteresting or inedible object in its environment, e.g., leaf-tailed geckos mimicking dead leaves, stick insects mimicking twigs. Chameleon characteristics versus other animals. Chameleons possess a sophisticated form of active camouflage that distinguishes them from many other camouflaged animals. Though they share some underlying principles with those capable of dynamic color change, like cephalopods. Chameleon specific characteristics and mechanisms. Chromatophores, like many other animals, chameleons have layers of chromatophores. Xanthophores/erythrophores, contain yellow/red pigments in the upper layers. Iridophores, these are crucial. They contain guanine nanocrystals in a lattice structure. By contracting or expanding the cells, chameleons can change the spacing between these nanocrystals. This alters how light is reflected, causing shifts in color, e.g., a tighter lattice reflects blue/green, while a looser lattice reflects longer wavelengths like yellow/orange/red. This is a form of structural coloration that allows for rapid shifts. Melanophores contain melanin pigment. These cells have branches that can extend over the other chromatophores. When melanin disperses, it darkens the overall color by obscuring the colors beneath. When it concentrates, the underlying colors become more visible. This mechanism fine-tunes brightness and contrast. Neural control. Chameleon color change is primarily controlled by their nervous system, allowing for rapid and precise responses to environmental cues, light, temperature, background texture, and internal states, mood, stress, mating displays. Purposes beyond camouflage. While camouflage is a key use, chameleons frequently change color for communication, signaling dominance, submission, or readiness to mate to other chameleons. Thermoregulation, darkening to absorb heat when cold, or lightening to reflect heat when hot. How do chameleons compare to other camouflaged animals? Shared characteristics with other active camouflagers. Chromatophores, cephalopods, octopuses, squid, cuttlefish also have incredibly sophisticated chromatophores, but their mechanism is different. Cephalopods use tiny muscles around each chromatophore to rapidly expand or contract pigment sacs, changing the visible surface area of the pigment. They also utilize iridophores and leucophores, which scatter all wavelengths of light, making them appear white. For even more complex patterns and textures, neural control. Like chameleons, cephalopod color change is largely under direct neural control, allowing for almost instantaneous changes. Multiple functions. Both chameleons and cephalopods use their color-changing abilities for camouflage, communication, and sometimes thermoregulation. Differences, compared to passive camouflagers, fixed versus dynamic. Animals like stick insects, snow leopards, or most fish, other than flounder or those listed previously, have a fixed coloration that provides camouflage. They cannot actively change their appearance beyond perhaps minor shifts in pigment distribution over a longer timeframe. Mechanisms. Passive camouflagers primarily rely on pigmentary coloration, biochromes, that are structurally embedded in their skin or fur, or on fixed structural colors. They lack the rapid, neurally controlled mechanisms of chameleons and cephalopods, adaptation versus response. Passive camouflagers are adapted to their typical environment. Chameleons and cephalopods respond to their immediate environment, while the goal of camouflage is the same across the animal kingdom, to hide. The science behind it varies greatly. Chameleons represent a peak in vertebrate active camouflage due to their unique combination of structural and pigmentary coloration under rapid neural control, allowing them a versatility that fixed-color animals don't possess.